All right, so it's January 20th, 2021, a historic day for two reasons. Number one, today is Inauguration Day, so our country is welcoming the 46th President of the United States. He was actually sworn in at this point about 15 minutes, 20 minutes ago, maybe even 30 minutes ago if you've been watching the inauguration.
I was watching some of it during lunch break.
Yeah. So it's a cool sight to see, not only because we are welcoming a new president, with that comes new policies, new politics, new ways of doing things, but also because of the historical nature of Kamala Harris being sworn in as the first woman, first Black person, and first Asian American to be sworn in as the Vice President of the United States. So it's obviously a huge deal, and there are a couple of other important reasons for this presidency that we can talk about some other time. And it's also a historic day because it is Sienna's birthday. So we can go ahead and wish them a happy birthday today in the chat, and hope that they are enjoying their birthday and doing all that they need to do to feel loved and feel grateful and enter this new year of life with a full head of steam. So congratulations, you've made it to another year. That's always a big deal and something to be happy about. But relevant to all of you all, we are going to cover nothing new today. We're going to cover some review topics, and we are going to talk a little bit about what the Unit 1 test will look like tomorrow, and we'll have some opportunities to practice. One thing I do want to say is that in order for you all to get your attendance today, you do need to participate in that live practice. So I want to discourage you from thinking about dipping out soon, or putting the computer down because your camera's not on and walking away to do something else. In order to get your attendance counted today, I do need you to participate in that practice, just so I can see if there are any blind spots that we need to go over before the test tomorrow. So the essential question today: what do I need to know in order to master the content from Unit 1, Biological Macromolecules? Does anyone remember what mastery is considered? What score do you need to get?
80 or above.
80 or above. So that's what we're all pushing for tomorrow. The test is not going to be super long tomorrow. There won't be that many questions. So in order for you to get an 80 or above, you're going to have to, get quite a few of them correct. Don't be discouraged, though. I think you all are well prepared, and you still have 24 hours to get prepared if you feel at the end of today that you are still confused about anything. And so there are several ways to do that. YouTube videos are a great way to do that, looking at some of our previous lessons. Reaching out to me one-on-one to ask any questions or maybe get some individualized tutoring, that's a great way to do it as well. Using a resource like Khan Academy or other YouTube videos, also a great way of doing it. Making sure that you are looking back at your notes. I encourage you all to do all of those things. In this first unit of biology, we've covered three standards, and that's exactly 10% of the standards that we will cover in this course. There are 30 essential standards in biology, the same 30 standards that apply to every high school student in the state of North Carolina. So this first unit, the first of 10 units, covers those first three standards. The first one is about organic molecules, standard 4.1.1. which states that students need to compare the structures and functions of those major biological molecules, carbohydrates, proteins, lipids, and nucleic acids, as related to the survival of living organisms. So we covered that really in the first few days of our Unit 1 studies. And I think you all understood it pretty well at the time, but I want to refresh your memory a little bit. The second standard is about DNA coding for proteins. So it's that relationship between DNA and proteins. Standard 4.1.2 states, summarize the relationship among DNA, proteins, and amino acids in carrying out the work of cells and how this is similar in all organisms. So we're thinking about the ways in which DNA code for proteins. Very, very important. We don't cover it too much in this first unit because we come back to it in the middle of the course and we talk a lot about it at that point. So we'll come back to this concept of the relationship between DNA, aka genes, and proteins, aka traits. The last of the three standards, standard 4.1.3, is about enzyme specificity. We talked a lot about enzymes over the last 2 days of class on Friday and yesterday, Tuesday. But standard 4.1.3 states students should explain how enzymes act as catalysts for biological reactions. So we're going to take a look at all of these things today. I'm going to be moving pretty quickly so we can get to the practice portion of class. And also, you won't see anything new. I literally copied and pasted the slides that we've gone over for the last week. So if you want to make sure that you can go in and write things that you may have missed, that's a good idea, but it won't be anything new. So we've got those 4 biological compounds: carbs, lipids, nucleic acids, and proteins. And I keep telling you about each of those 4 biological compounds. You need to remember 4 things: the monomer that makes up the compound, the elements that make up the compound, the function of the compound, and then you need to have some examples filed away in your brain of the compound as well. So behind box 1, carbohydrates, the monomer of a carb is called monosaccharide. Mono meaning 1, saccharide meaning sugar. So monosaccharide is 1 sugar. Another name for it, at least as it relates to this class, is glucose. So glucose we can consider to be the monomer of carbohydrates as well. There are 3 elements, the same 3 elements that appear in all 4 compounds: carbon, hydrogen, and oxygen. But the cool thing about carbs is that they appear in a 1:2:1 ratio. So that means that if I've got 8 carbon atoms, that it means I also have 8 oxygen atoms, excuse me, and I have 16 hydrogen atoms. If I've got 32 hydrogen atoms, that means that I have 16 carbon atoms and 16 oxygen atoms. That's what we mean by a 1:2:1 ratio. The function of a carb is to store short-term energy, which can be used quickly. We've got to have quick access to the energy, and carbs also contribute directly to the structure of some specific cell types, which we'll talk about in the next unit. And then our examples: carbs are sugars. So when we see words that end in OSE, fructose, sucrose, glucose, lactose, cellulose, galactose, these are all sugars. They're all carbohydrates. Okay, so that means that they all have that same monosaccharide monomer. And then also we threw in a couple other examples there as well: glycogen and starch. Lipids have the monomer of glycerol and 3 fatty acids. They have those same 3 chemicals: carbon, hydrogen, and oxygen. They need to store energy for long-term use. So we don't expect to use energy immediately from lipids. We expect to have access to that over longer periods of time. And this is in many ways why we think about lipids being in animals that hibernate. They don't eat for months at a time, but they've got these reservoirs of fat that allow them to still maintain, still survive, even though they're not actually eating anything. Okay, lipids, as we know, also provide insulation, as well as they make up the cell membrane, and that's important. We'll talk more about that in Unit 2, but we do need to know that for tomorrow's exam. Examples of lipids include steroids, phospholipids, waxes, oils, and fats. The third biological compound is called a nucleic acid. Nucleic acids are made up of nucleotides, which themselves have 3 parts: a 5-carbon sugar called pentose. Again, there's that OSE ending, a phosphate group, and a nitrogen base. The elements that make up a nucleic acid are carbon, hydrogen, and oxygen, yet again, as well as nitrogen and phosphorus. The function of a nucleic acid is to do really 3 things that are all obviously connected. Number 1, to store the genetic material. Number 2, to control which proteins are produced. And number 3, to control all of the cell's activities, meaning who gets to come in, who gets to go out, what time is this done, how many times do I do this, when do I reproduce, Do I need to grow? Do I need to shrink? Do I need to enter what's called programmed cell death? All of these things are decided by these nucleic acids in a special region of the cell, which we'll talk about in Unit 2 also. And lastly, examples, there are only two examples, DNA and RNA. Now there are some sub-nucleic acids, a subclass or subset of nucleic acids under those two more general types. But for the purposes of this class, we really just need to focus on DNA and RNA. Proteins are our last organic compound. The monomer of a protein is called an amino acid. We've got 20 different amino acids that can be arranged in different ways. They have different chemical properties, all of which determine the shape of the protein and the function of the protein. Elements that make up a nucleic acid: carbon, hydrogen, oxygen yet again, and this time we also add nitrogen. The function of proteins varies heavily. They do almost everything for the cell. We kind of call them the do-it-all organic compound. But we can think of some specific examples. As enzymes, proteins speed up reactions. As hemoglobin, proteins carry oxygen in blood. As insulin, proteins signal the breakdown of sugars. As antibodies, proteins help the immune response. And as hormones, they serve as chemical messengers. Okay, so hormones get transferred from your brain to your reproductive system, for example, or from your kidneys to your brain, carrying messages, telling different parts of the body what to do. So this is that important table. If you can remember this table, memorize this table, and be able to apply what this table states, you will be in a great position to master tomorrow's exam and to have mastered Unit 1. All of those asynchronous assignments that we've assigned over the last week and a half have been geared towards this table, essentially. Okay, so if you want to go back and redo some of them or make sure you've done all of your missing work, keep in mind all assignments, are due on Friday at 11:59 p.m., but they should also really be done before tomorrow because they provide excellent practice for the exam. So take a quick screenshot, a picture of this table if you don't already have one, or if it isn't already written down in your notes. I'll leave it up for just a few more seconds. All right. Now we ran through this mind map as well, but I'm going to go through this one more time pretty quickly. The stream on the left, proteins, which are made of amino acids. Examples include enzymes and hemoglobin, antibodies, insulin, and hormones, as we just said. So I like mind maps like this. They allow you to add on as you learn more information, make fluid connections. You can draw arrows between things that are not already connected. For example, you might want to draw an arrow between DNA, which would be in this circle, and proteins, just to give yourself that mental reminder that the relationship exists. That second stream, carbohydrates, which are made of monosaccharides, also known as glucose and cellulose. I'm going quickly, as we can see, because we've already seen this slide and I've asked you all to answer these questions before. Lipids, which are made of glycerol and 3 fatty acids, whose jobs include long-term energy storage, as well as insulation. And our last stream on the far right, nucleic acids, which are made of nucleotides. The two main types are DNA, RNA, and these lead to genes, which then lead to proteins. So I'll leave this up for a few seconds as well so folks can grab pictures. I really, really encourage you to draw mind maps. You can start off small, then you might want to staple a couple papers together on either side and keep adding on and get bigger and bigger and bigger. And you'll, at the end of the course, have this giant visual representation of all of the concepts we've introduced this year. I think that would be excellent. All right. The relationship between DNA and protein. So this is that second standard that I mentioned. I won't talk too much about this again because it comes up really in units 4 and 5 of the course, and it will become a much more important theme later when we introduce some more complicated concepts. But we can see that the relationship exists. So nucleotides are the monomers of nucleic acids, like DNA. So nucleotides combine to make DNA. And remember, the order of the nucleotides determines what message the DNA carries. Just like the order of words in a sentence determines the message of that sentence. My name is Mr. Rudd carries a specific message. I can switch the order and say Mr. Rudd is my name. That says basically the same thing. Rudd is my Mr. name. That doesn't really sound right. Okay, so when you start switching the order of things, whether it be in a sentence and you're switching the order of words, or whether it be in a nucleic acid and you're switching the order of nucleotides, it starts to get a little fishy, right? So there has to be a specific order to carry a specific message, and that message determines which amino acids are brought into the picture. And then the amino acids get linked and they become proteins. So that's the relationship. The DNA is carrying a specific message, which tells the cell, I need this amino acid, I need that amino acid, I need that one, and I'm going to link them together in that specific order. The order is very, very important. And as we know, shape determines function. All right. And then that third standard was all about the specificity of enzymes. And we've talked about this before. You've seen this slide for the last 2 days of class, and you're seeing it for a third time. But in review, enzymes are 3 things. It's a typo. I hate typos. Okay, though. Number 1, they are catalysts. They speed reactions up. And we're going to talk about how they do that, but I know you all already have some thoughts because you wrote it in your notes yesterday. Number 2, enzymes are specific. They don't just work with everybody. They can only work with specific substrates, just like a lock and a key. And I think it was Ashley who said that yesterday, a lock and a key. And number 3, enzymes are reusable. So they remain unchanged throughout a reaction. You can put them in and they should come out the exact same way. Oh my gosh, I should do it again. Okay. Cool. So the way that an enzyme speeds up a reaction, the way that it serves as a catalyst is by lowering the activation energy of the reaction. So every reaction has a certain activation energy, and that is the amount of energy that is needed just to start the reaction. There's always some amount of energy that is needed in order for the reaction to take place. Enzymes lower that amount, making it easier for the reaction to take place and faster for it to take place as well. So we can look at this image from yesterday, and we can see that the reactions, or the hills, are different sizes. The hill that represents the reaction without an enzyme has a much higher peak. It's got a higher activation energy. The hill that represents the reaction with an enzyme has a lower peak, representing a lower activation energy, which means that that green hill is going to represent a reaction that goes faster. All right. So again, reactions can and will happen without enzymes, but they are sped up by enzymes to make them really more feasible for living organisms. Can't wait all day for every reaction to take place. We need them to happen very, very quickly in some cases. We talked about this as well. I showed you guys how enzymes are reusable. We've got the enzyme. We can see the enzyme's active site here, which is where the reaction actually takes place. We can see a substrate that fits perfectly within this active site. And of course, within a given cell or within a given environment, there will be plenty of different substrates, not just one. And so the enzyme will first act on one substrate and turn it into products, but then it's going to move to a different part in the cell where it can act on another substrate and turn that one into products, and then it'll move somewhere else. So all we're doing is setting off this series of chemical reactions, but all the while the enzyme is not being changed. If everything is, if the conditions are right, the shape of the enzyme should not change, which means that it should be able to continue to act on more and more substrates and create more and more products. We talked about this yesterday, but I had to break it down to my other class earlier today. If I've got a Twix bar and I snap it in half, I don't now have two Snickers bars. I just have two smaller sized Twix bars. If I've got a Skittle and I cut it in half, I don't now have two M&Ms. So when I break something down, it becomes its own parts. It doesn't become something else. It doesn't become parts of something else. When I break a protein down, it becomes amino acids, because amino acids are the monomers of proteins. If I break down a carb, I don't end up with nucleotides. Okay, nucleotides are not the monomers of carbohydrates. So when I break down a carb, I end up with monosaccharides, which are the monomer of carbohydrates. Deoxyribonuclease is an enzyme that breaks down nucleic acids into nucleotides. And lastly, lipase is an enzyme that breaks down lipids into glycerol and free fatty acids. We notice that all of these words end in ASE. So an ending of ASE indicates an enzyme, which of course is a protein, and an ending of OSE indicates a carbohydrate, or a sugar, which is of course a carbohydrate. So again, we want to have those mental cues. Start making those connections. That mind map is a great way to do that. If you're presented with a little bit of information on that mind map, now you've got this visual representation of all the different information that branches out from that. So if I just give you that a word ends in ASE and you've never seen the word before, well, then you at least know that ASE means that it is probably an enzyme, which means that it's a protein, which means that it's made up of amino acid. It also means that it is a catalyst. It also means it's reusable. That also means it's specific. But that also means it has an active site. And it also means that it can be denatured. So just with a little bit of information, now you've got all these other thoughts branching through your brain because you've been able to make good connections. That's how you become a good test taker. That's how you become a good student. And most importantly, that's how you become a strong learner. We also talked about enzyme function and the fact that enzymes have each enzyme has an ideal environment. Some classes of enzymes prefer hotter and more acidic environments. Some classes of enzymes prefer cooler, but still acidic environments. Now, if they're placed in an unfavorable environment, they become what is called denatured. And to be denatured means that the shape of the active site has been damaged and it no longer fits with its designed substrate, with its designated substrate. So when we look at enzyme graphs like this, we should know that they typically represent enzyme activity. So as we move up on the y-axis, we're going up this graph. What we're saying is that the enzyme activity is increasing. That means the enzyme is working better and better and better. And then it gets to a peak, and then the enzyme activity starts to fall off. The enzyme isn't working as well. This peak is going to represent what's called an optimum environment. In this case, it represents an optimum pH. So we saw this curve, this graph yesterday, and we found out that the optimum pH here is 8, as indicated by this dotted blue line. Obviously, on your test, you will not have a dotted blue line, but you'll have to kind of, see it yourself. On the other hand, we talked about the fact that when an enzyme is denatured, that means that its shape has been changed and it's no longer working. So here, when the enzyme activity is at 0, we're on the x-axis. That means that the enzyme obviously is not working. In this case, it's happening in 2 places. Anything less than about 4.8 or 4.75, the enzyme is not going to work. And anything greater than about 10.5, again, the enzyme is not working. So at those pHs, that enzyme will not function. On the other hand, we've got another enzyme. This time we're seeing the effect of temperature on the enzyme. So again, there's an optimum temperature. The enzyme activity is increasing until we get to a certain peak, and then it falls off again. When the enzyme activity is at 0, of course, that represents that the enzyme has been denatured. Its shape has changed to the point where it no longer functions. The optimum temperature here, again represented by this dotted blue line, is clearly telling us that it is 40 degrees Celsius. And the denatured temperature, we can't really see what happens when the temperature goes negative. But we can see that anything above 62 or 63 degrees Celsius will represent denaturation. All right, so at this point, we're basically making perfect timing. Now I want to do the practice portion. So we are going to do a Nearpod. I'm sure you all have done these before. I'm going to send the link to the chat for you all to go ahead and join. As soon as you join, it should allow you to start engaging in the activities. The first activity is a matching activity, and you'll need to match the following terms with their pairs. So again, this is how you get your attendance for the day. I want to see you moving through these different questions. I just sent a link to the chat. All you'll need to do is click on that link, or you can go to join.nearpod.com and enter the code 6K5ZB. I also just sent that to the chat. Thank you, Lance, for joining. Thank you, Janelle. Thank you, Melissa and Ashley and Lisa. All of these folks will be given, well, you got to make your way through the Nearpod, but you're on the right track to getting attendance taken. Thank you, Araceli and Lisa and Abigail and JT. So we've got 8 people so far, but we've got 17 people in the Google Meets. So I need to see a lot more people joining. 9 more of you have to join up. Thank you, Dijon. So we've got 8 more people need to join. Thank you, Josue. And I can see Lance, Melissa, Janelle, Ashley, and Lisa have all made progress. So those of you who have joined, go ahead and start matching those terms up. 7 of you still have yet to join. Let's go ahead and do that. Abina, where are we? Ashanti, where are we? Ashley, oh no, you're in there. Carolyn, if you're having difficulties, please let me know. Lance has finished this first section, and everybody who's here has joined. Okay, Abina, thank you for letting me know. So, did you try clicking on the link? I'll send it one more time. Nico, Santuan, Siana, Tyshawn, all of you all should be joining. Thank you, Ashanti. Okay, Ashley's done. Abigail is done. Okay, so go to join.nearpod.com. Abina. Okay, good, it works. I guess you're entering your name, and yep, you're in there now. Good. So we've got 11 people. Six more of you. Nico, where are you? Siana, Santuan, that's 3 of the 6. Carolyn. Thank you, Tyshawn. 4 folks still not in. It says, did you try to click? Just all you really need to do is click on the link, but make sure you've got the code typed correctly. It might need to be, oh, I'm sorry, it should be 6K5 ZD, not 7K5 ZD. My apologies. Cool. Okay, we've got 15 out of 17 people. Okay, I'm missing Siana, and I'm missing Carolyn, I think. Siana and Carolyn. So hopefully it lets you just go ahead and get started on that quiz. We had some problems with this in my last class, but I think you all should be able to just get started. Okay, thank you for letting me know. I can see that Dejan has started his 8-question quiz. That first question is a little tricky because there are 2 correct answers. I don't know if you need to click both correct answers or if I think you might need to try to click both. Yeah.
What is albumin?
You don't really need to know exactly what it is. All it says in the question is it's a protein that's found in egg whites. So this is a good test taking strategy, and I'm glad you brought this up. When you see a word that you don't really know and you've never seen in our class before, it likely is just there kind of as a distractor. So there's another word in that sentence, that first sentence, that really is all you need to know to answer the question. Says albumin is a protein found in egg whites. Okay, so I guess I'm still learning Nearpod a little bit, but as I switch through these different quizzes or these different activities, it forces you guys to switch back and forth. So what I'm going to do, I'll leave it here on the matching pairs for 2 more minutes, and then we'll go back to the quiz so people can finish their quizzes, and then we'll move on after that.
I already did both activities. Do I need to redo it?
No, if you finished the activities, then there's no need to redo them, but I just want to give everybody else who came in a little bit later an opportunity to get caught up. And there's one more, there are a few more activities. There are 2 polls that I want to take, and then there's a fill in the blanks activity as well. Okay, I'm moving on from the matching, and let's see if we can finish up this quiz. It looks like I can see that 6 of you have finished it. We've got 16 people here, but it looks like 3 people haven't even started. So let's give this about 4 more minutes until 105. Okay, I can see that it looks like everybody who is trying the quiz has done it. So I'm going to move on now. This next one is a live poll. Actually, let me just go back really quickly. I'm sorry. And I want to share the results. So hopefully you all can see the results of the quiz. Did quite well. So I'm definitely proud of you guys. Let's see. Melissa got every question right. Abigail got every question right. And Carolyn got every question right. And several of you only missed one or two. So that's definitely a good sign. Proud of you guys. Let's move on here. Big brand moment, yeah. All right, so the question here that I just want you all to quickly answer, how confident do you feel about your ability to master tomorrow's exam? And like Lance reminded us, and I think JT chimed in as well, mastery means earning an 80% or above. Okay, we're still waiting on some answers from people. Just go ahead and just quickly answer something. Uh, I need an answer from JT. I need an answer from Dejan, Shanti, Tyshawn, Santuan, and Nico, if you're still here. We need some quick answers just so we can move on to the next question. 30 more seconds. Okay, don't want to spend too much time. Here are the results of this. So uh, we can see that about a little over half of us feel somewhat confident, a quarter of us feel very confident, and then no one is feeling nervous. We've got several people who did not answer. But if you're feeling somewhat confident, or if you're feeling very confident, that's excellent. Still prepare as if you are feeling nervous. Set yourself up to do quite well. Prepare by checking over your notes tonight. And I don't want you all to be discouraged if this first exam does not come out how you want it to. Our exams in biology are hard because the EOC exam is pretty hard. So we need to prepare you all and get you used to the quality of questions and the difficulty of the questions that you'll see. Okay, another question. Which topic are you most concerned or confused about as it relates to our first unit of the course? So tomorrow we'll take a little bit of time to go over one last thing, and I want to know what makes sense to go over. Does it make sense to go over our 4 biomolecules, which would be answer choice A, the relationship between nucleic acids and proteins, which would be choice B, or enzymes, which would be choice C? So go ahead and answer that for me really quickly as well. Okay, let's get a few more. We've only got, looks like 3 people who have not yet answered. Abigail, Santuan, and Nico. You all can just give us an answer. It looks so far like most people are saying enzymes. They want to talk more about enzymes, so we can definitely go over that tomorrow. Okay. So here are the results. Half of us said enzymes. Almost the other half said the relationship between DNA and proteins. Keep in mind that that is a relationship that we will explore a lot more throughout the course. That's why we don't spend too much time in the first unit with it. But I can go over that a little bit tomorrow before the exam. So thank you all. One more thing. This is a fill in the blank activity. So I'm not exactly sure what it will look like. But hopefully there's a paragraph and then there's also a word bank, and you all just need to choose the words that fit in with each sentence. Let me take a look at the student view really quickly. Yeah, so it looks like a drag and drop type of thing. So I think, Janelle, from what I can tell looking at the student view, there should be a paragraph there on your screen. And there should be some words missing from that paragraph. But at the bottom below here, what's that?
There is no paragraph. It's only the blanks.
Oh, really? You can't see the words?
I mean, there's the dragging words that are on the bottom.
Yeah, I'm not sure why the paragraph itself is not showing
I can
up.
show you my screen if you would.
Sure, why don't we do that?
This.
That's not what it's supposed to look like. Okay, well, I'm not sure how to fix it right now.
What does it look like on your screen?
Yeah, let me show you that. I can see that Josue, it looks like it worked for Josue. He got everyone correct. Okay, so this is what it should look like. You can see a paragraph and certain words missing. So which should go in this first box from the word box below? An enzyme is called a blank because it helps to blank chemical reactions, which should be in this first box. Any ideas? If you all can see my screen.
A catalyst.
Good. Catalyst should be first because it helps to speed up chemical reactions. An enzyme is able to speed up chemical reactions by lowering the blank energy.
Activate.
Good. The activation energy of this reaction, which is the amount of energy needed for a reaction to take place. Without enzymes, reactions would be much
slower.
An enzyme is said to be specific because it will only work with blank that have a specific blank. Any ideas?
The second one is function.
You can think about putting, so you could probably put function there, but I'm thinking of something else that is very closely related to function. Blank determines function. That should give you a hint. What do I keep saying? Blank determines function. Good, thank you. So this box is shape. This box is function. Shape determines function. We're still missing a box, but we can kind of do process of elimination. The blank site of the enzyme must fit with the shape of the reactant. So what is this blank here? Okay, yeah, you're right. Thank you, Janelle. Substrates should be up here. And what should go here? The blank site of the enzyme must fit with the shape of the reactant. We've only got 6 choices remaining. Thank you, Janelle. The active site of the enzyme. If the shape of the enzyme changes, the enzyme is said to be blank. This means that the enzyme is no longer going to function properly. Thank you, Josue. Denatured should go here. An enzyme has an ideal environment in which it works best. And this is determined by conditions such as blank, how acidic the environment is, and good, thank you, Ashley, pH, and blank, how much heat is present. Excellent, thank you. Lastly, enzymes are blank because they can be used more than blank.
Reusable once.
Excellent, thank you.
On my screen, it's not giving me all the options.
You know, we'll keep this in mind. I just won't use this aspect of Nearpod next time. But I appreciate you all sticking with me. So let me actually share another screen really quickly as we close out class today. And I appreciate everybody participating. So there is an asynchronous assignment. It's a practice quiz with, excuse me, I can't talk, focusing specifically on enzymes. So those of you who answered that you wanted some more help with enzymes, that's going to be a great place. You know, the best help comes from doing yourself. And even if you don't do well, at least you've kind of exposed yourself more to the idea. Keep in mind, all of the assignments from Unit 1 will be due Friday, but we will take Friday to do some one-on-one conferences so I can check in on you all. And then also for you all to kind of have it as a work day to get your missing assignments turned in and get full credit for your Unit 1 assignments. Then there's also that Biomolecules and Healthy Living Creative project that is only for this class, only for my honors class. So keep that in mind as well. It might seem like a lot, but hopefully you've been kind of staying up to date with things as we go. We've got the exam tomorrow. And take some time today to take in the inauguration and to kind of absorb the moment. This is a historic moment. The next time we elect a president, you all might be eligible to vote. So that's something cool to keep in mind. But other than that, study, and I will talk to you all tomorrow.
See you tomorrow.
See ya.